Hello, everybody. My name is Marie, and I'm going to be talking about inhibitory postsynaptic potentials and excitatory postsynaptic potentials. I would like to begin by talking about the anatomy of a synapse. And recall that that synapse is the space in between two neurons. So a neuron is really getting contacts from many presynaptic inputs, and it's also making connections at the postsynaptic end with many neurons. So if we look here on the left, you can see that those dendrites are receiving information all onto this one cell body. And then the cell can actually have connections with many postsynaptic neurons down here toward the bottom. And if we look at the space in between two neurons, there are two ways that they can be either joined or not joined. On the right, they have cell adhesion molecules that are kind of tethering and anchoring the two cells together. Whereas on the left, there is this space in between, which is like an ocean of space because electrical currents cannot really make that jump across. So neurotransmitters play the role of bringing that information, and it's only when they fit like a lock and a key, these chemically gated channels, is when they will have an effect on the postsynaptic neurons. So on the top, you can think of this as being the presynaptic neuron, and then the postsynaptic is on the bottom. And these ionotropic receptors are ligand-gated ion channels. These are all chemically gated channels. They are not voltage-gated channels. So we are not talking about really getting action potentials here necessarily. Metabotropic receptors here simply mean that the ligand is going to make a connection here, and then there's going to be some metabolic pathway that will travel on and have an effect on the neuron. The postsynaptic neuron, you may come across the word subsynaptic cleft, and I don't want that to be a source of confusion. Just think of that as being the postsynaptic one. That's just simply a biological word for this area just underneath the surface here. So that's the subsynaptic cleft. Generally, information is going one way, and we can get different kinds of potentials. One of two things can happen. Either the neurotransmitters are going to cause this graph to go up or down, which means it's going to make that potential either more positive or negative, one or the other. So if it becomes more positive, that is an excitatory postsynaptic potential starting here on the left, and that is heading toward threshold. So it's becoming more positive. Notice that it does not quite get all the way to the threshold. It may or may not. And the inhibitory postsynaptic potential, which is permeable to potassium and chlorine ions, has a hyperpolarizing effect, and that's going the opposite way. So it's really the interplay between these two kinds of potentials that our nervous system integrates all the information that is coming in. But we're talking about two different kinds of ions for each of these potentials. They come in varying magnitudes, and it's only when they hit the threshold that you will actually have an action potential. Many factors influence the magnitude of either the EPSPs or the IPSPs, and I just want to take a minute to mention neurotransmitters because they matter. They have varying effects, and then we're going to talk about temporal and spatial summation. This slide I included just as a reference because many of these neurotransmitters actually are become familiar to us as we think about various drugs. For instance, acetylcholine is a neurotransmitter that acts much like nicotine, or I should say nicotine acts like acetylcholine. It has a physiological and psychological effect and it binds to the same receptor and causes excitatory postsynaptic potentials. Botulism is a toxin that inhibits presynaptic release of acetylcholine, and we all know Botox as being a drug that actually, a toxin that affects facial muscles and used for cosmetic surgery. Dopamine, there is thought to be a lack of dopamine in Parkinson's disease. Serotonin, Prozac is a drug that enhances serotonin by inhibiting its reuptake. And gamma aminobutyric acid, also known as GABA, that is one where the drug Valium, diazepam, binds to GABA receptors and causes inhibitory postsynaptic potentials, which means it kind of it diminishes excitatory ones and is used for anxiety. So in general, the same neurotransmitter is generally released from any particular presynaptic neuron, and they generally have the same effect. Summation is the idea that you can get an additive effect from many excitatory and inhibitory potentials. And here we have on the left a graph showing an actual neuron and a graph showing how you can have multiple excitatory and inhibitory potentials having some effect. So notice up here in this second diagram, we have excitatory ones coming in, and there's an inhibitory one in red. And temporal means tempo. Think of it as being time. And if you're getting these impulses and these postsynaptic potentials coming in, we're talking microseconds apart, many of them can actually add up in time and have an effect. These potentials are coming across the soma, the body of the neuron, and they actually, depending on where exactly they come in, they will diffuse across, and their effect is actually weakened because really molecules are running out. So if it reaches the axon hillock, then that will be the determination as to whether an action potential is fired off. And of course, it has to hit that threshold value of about 50 to 55 here, as you can see on the graph. Spatial summation refers to space, or the area in which you're getting these postsynaptic potentials. So here they're coming from very different areas, and if you get many of them, they can actually add up to causing an action potential. And here, I just want to remind people that you're getting many of them all the time. And again, it's this integration of how many we're getting and where we're getting them from for each of these neurons that's going to have some effect. Down here, I just included a little link here. It was just a very good little review of EPSPs and IPSPs, and you can look at that if you need to. So one study that was particularly interesting was one done by Walter Paulus and John Rothwell. And this is a paper from 2016 called Membrane Resistance and Shunting Inhibition, where biophysics meets state-dependent human physiology. And it's really a meta-analysis of many studies where they were looking at whether the activity in the brain is changing and what kinds of influences are having an impact on membrane resistance. So the activation of neurons changes membrane potential and also reduces membrane resistance. And if you reduce membrane resistance, then you really are increasing conductance. And they think that this happens in shunting inhibition. And when you shunt something in an electrical circuit, it really just means that you're providing a way to join 2 points, and the current is going to be diverted. So shunting inhibition It actually, they believe, reduces somatic depolarization. It increases synaptic conductance, and there's no real obvious change in the membrane potential, but this seems to be a mechanism that's going on that's going to influence how resistant or how conductive a neuron actually is. And in this diagram, you can see the current is coming here from the right, and these are excitatory pathways, whereas these are inhibitory pathways. And then you end up with this outward current flow here, where increase of conductance is being caused by this shunting over here. So their hypothesis was really just that this transmembrane resistance plays a role, and they are using transcranial direct current stimulation and transcranial magnetic stimulation to see what these neurons are doing. They also wanted to see whether the person being active or voluntary action, or if the person at rest, if the neurons are at rest, actually plays a role. And they wanted to see what that change in response would be. Here is data that they provided in their paper, and these are a series of time courses of postsynaptic conductances. And if you look on top here, this is a super simplified version of a neuron, and this would be the soma, the cell body here, and this would be the dendrite. This is excitatory, this little black bubble coming in, and the red is an inhibitory synapse. And if you look at these graphs, what they're showing is what is happening with the synaptic reversal potential and the resting potential. So when we look at this, the first one really is a control where it's just showing a series of action potentials. The excitatory synapse is activated multiple times. You can see it across here. And really, this x-axis would be time in milliseconds, and this, of course, would be membrane potential with about minus 50 up here. So when we look at this, for instance, this first one, the resting potential is more negative than the chloride ion potential, and there seems to be a reversal here. It's actually, they're looking at GABA receptors, and the chlorine channels are opening, and what's happening here is they're actually flooding in, and uncharacteristically, there's a depolarization. Usually we think of GABA as being a hyperpolarizing or repolarizing activity. In this one here, and with really all of these, you can be looking at these little red lines here. The red means that the inhibitory potential is activated. The black means that the inhibitory is inactive. You can see here where the synaptic reversal potential is equal to the resting potential, you're not really getting any EPSPs or IPSPs. And they think that shunting inhibition doesn't produce significant depolarizing or hyperpolarizing currents. And then down here, the resting potential is greater than the synaptic reversal, and there's less negativity than the chlorine equilibrium potential. There's an activation of the inhibitory response. The synapse generates an IPSP, and then this leads them to believe that the shunting inhibition will add some effects and increase the effectiveness of the neuron. And you may be asking why any of this is very important. These scientists do work with transcranial stimulation. This TS research is really important for conditions like depression. This last slide are my references, with the main one being the paper by Paulus and Rothwell. And thank you.